So most people think about lion's mane mushroom and they think it's, one species, one medicinal profile, one source of erinosins and hericenones and all the interesting bioactive molecules. But that assumption is, it's at very least very simplified. There's multiple hidden species with different genetics, different chemistry, different life histories. And that's the rabbit hole you went down, Julien, right? During your master's thesis under Greg Thorne. And it started off kind of, well, and you correct me if I'm wrong, but it started off as a taxonomy project, but then you kind of brought in some biofermentation and, medicinal bioprospecting and then evolutionary chemistry and fermentation science. And that's kind of how it evolved, right?
Yeah, absolutely. Initially, it was going to be a targeted analysis for bioactive metabolites produced by Hericium. And then as we got into the work, we realized, oh, there's some taxonomy work that needs to be done with this genus. And I was in Greg Thorne's lab. He is a fungal taxonomist. So we decided to tackle that problem as well. So in the early phases, it was just kind of, let's see what species are out there. Let's see how we can separate them and delineate them and actually put a name to some things. And then from there, we'll be able to have a more targeted analysis of what these species produce in their mycelium. I was more focused on erinosine A. That's a terpenoid that's produced in the mycelium of the fungus. It shows nerve growth factor potentiation in a bunch of studies that we took, and we kind of rolled with that aspect of things. There is some fruiting body metabolites.
Yeah, so the hericenones.
Yeah, exactly. The hericenones are also super interesting. We didn't go that route primarily because I didn't want to spend the time fruiting the mushroom.
Fruiting them.
And yeah, liquid culture is just so much easier to work with. But that's kind of where we ended up with it.
Yeah, really cool. So your work, I mean, it resolved some of the confusion. I mean, there's a bunch of confusion in the space, but it came up with some conclusions around the species complex. And it also looked at the different lineages of Hericium, not, the ones not called lion's mane, bear's head. They all have different names, but like the Hericium colloides, Hericium, well, let's list the five. So it's Hericium colloides, Hericium americanum, Hericium, well, now you got organensis. Is that
organensis on the west coast and Carolinus
on the west coast,
on the east?
Carolinus on the east coast and up into like southern Ontario, and then Hericium carathum. I'm probably butchering that.
Is how I say it.
Ceratum. Ceratum is that. I've heard other people say that once you said it, so that I'm sure that's it. And that's more of a montane species. And I think you got your sample out of Alberta somewhere, right?
That's right.
And,
yeah,
and
that's
I've
a
seen
rare species. We were lucky to get a sample out of there. There's not many observations on INAT even. And the last one is Abiatus, which is on the west coast.
Yeah, which is the conifer species on the west coast,
That's
right?
Yeah.
Yeah, and I've actually eaten that one. I live on the west coast, and it's probably the one I find the most, mostly at elevation, close to like it seems to really like mountain hemlock out here. So wherever there's mountain hemlock, you can find it quite readily in the summer.
Oh, man, I'm jealous.
So is that 6, or did I overlist?
That should be 6.
Well, I guess we're grouping the two Hericium carolensis and Hericium organensis under the Hericium erinaceus complex, right?
Yeah. That's how we.
Although you are proposing them as their own distinct species, is that correct?
That's right. Yeah. Both in the complex erinaceus, but distinct species.
Interesting. Yeah, and that's really that's the big blockbuster, because all this work's being done around Hericium erinaceus, but it's probably a lot of it. I mean, all the Japanese work in the 90s that identified these diterpenoids, the erinosins. It was probably using. I mean, a lot of it's pretty vague where the culture came from, but it's probably using the local Hericium erinaceus species, which turns out to be Hericium Asiatum, or you pronounce it better than me, I'm sure. But,
yeah. Well, we decided on Asiaticum for the species that occurs in the type specimens from Japan, but there's probably
more there.
Around. Yeah, and a lot of the early work was done with then known erinaceus. Now,
which is a European
species, is Asiaticum.
Yeah. And so we know what we're calling the true Hericium erinaceus, or the species that holds that true name, is actually a European species, right? Because that's where it was first described. but likely a lot of this Japanese work wasn't using that species, right? It was using the Asian species.
For sure, yeah. And you know, species names, species concepts, these are all our like we impose these on
Yeah,
these
human,
organisms.
they're
It
human
doesn't
construct.
the organisms
Yeah.
don't care if they're called one name or another. They're
No.
happy to go about their lives doing their
Yeah.
thing. But yeah,
Yeah.
in terms of the literature and the kind of academic treatment of these species, it gets a little bit more nuanced when we apply certain names to what species in order to track their lineage, which does it does have an effect when we are breeding mushrooms or listing them for
Well, it matters.
purposes.
It matters to the chemistry too, right? Because the different life histories potentially evolve different chemistries. And if you're going, oh, look at this chemistry in this population of fungi, it doesn't mean a population from halfway around the world is going to have the same chemistry, right?
That's right, yeah. But interestingly, in our study, we found that so we collected all these strains. I collected a lot of them myself, but we also got cultures from around the world, and we tested them for erinacine A production, and all of them showed production of erinacine A. So, at least with respect to erinacine A, it's probably a genus-wide phenomenon. I can't say that for
Yeah.
sure because I didn't test all the species in the
Yeah.
genus,
Yeah. Yeah.
but it looks to be that way.
Yeah, really fast. I mean, my biggest interest in this molecule is like, why the heck are these mushrooms all producing this? I mean, they're obviously putting a significant amount of energy in it. It seems to create neuronal differentiation in mammalian brains, but there's no way it evolved to do that. I mean, it's basically it's a saprobic fungi, but it's also parasitic, right? It infects living trees. So, my guess would be it's interacting with the host, it's interacting with itself, an it's a competitive advantage. But is there any insight into what these things actually do?
I mean, we can speculate, but we can't.
Yeah, well,
At least
that's what we're here for, right?
in terms of studies that have like proven the function of erinacines in these organisms, there hasn't been any
Nothing.
definite conclusions. I tend to believe that yeah, the mycelium is interacting with the host and other microbes competing for that substrate in it's saprotrophic. It's competing with all kinds of organisms to capitalize
Tons.
on the
Every
nutrition.
Yeah.
It might be an antimicrobial compound. It might have some other effects, but we can't say for sure. But yeah, you're right. This is not an this is an expensive compound to produce. It's not like they evolved this thing and put energy in towards it just for
For no
fun of it,
reason.
or just for supplement companies to sell it to you.
No, yeah. Yeah.
But terpenoids are in plants at least. They're often used as herbivory or anti-herbivory compounds or defense compounds. So it stands to reason that it might be doing a similar thing for the Hericium species that are producing it as well.
Yeah. Well, I mean, it's in the mycelium too, which is like the growth phase of the fungus, right? That lasts for years, which it's fascinating. It doesn't seem to be particularly toxic though, right? But maybe it is to competitor fungi or bacteria or something. There has been some antibacterial work, I think, on it. I don't think it was, particularly antibacterial, at least to the pathogens they looked at, but maybe it is to something in the tree.
Yeah.
Yeah, interesting. It's fascinating. It's just the mechanism of action, like it binding to the neurite receptors and causing neurogenesis. You almost wonder if it has some sort of growth effect in the fungi, but I mean, I guess that somebody else has to figure that one out.
That's someone else's PhD to do, not mine.
Yeah, exactly. So let's talk about your background a little bit. Were you into mycology prior to this project, or were you already a myco nerd?
I was more of a plants guy.
Okay.
In my undergrad, I was doing more plant science stuff, and then after I graduated, I was handed a book about mycology from a friend, and that kind of just turned the lights on for me. I was like, oh, this is it right now. There wasn't a whole lot of mycology courses available where I did my undergrad, but then I discovered mycology, fell in love, and sought this out afterwards, and yeah, was fortunate enough to find a spot in Greg Thorne's lab, and then I've
Yeah,
been working
he's
with mushrooms
renowned
ever
fungal
since.
systemicist, right?
Yeah.
Really cool lab to be part of. And so his but you came into it actually more interested in the in like just studying what bioactives these things are producing than the taxonomy. And so I told it backwards in the beginning. It started as a bioactive search and went into taxonomy when you realized there was a mistake or
Yeah,
like just some confusion, I guess, as opposed to
totally.
a
Yeah,
mistake.
I kind of was going into the project thinking it would be primarily about, industrial applications or bioactive production of different metabolites. And then Greg kind of looped me into the taxonomy world and taught me
Yeah.
about taxonomy and
Yeah.
sequencing and genetics. And I was, I guess this is part of the project now.
Cool. Well, but it's a broad background. I mean, you touched on so many different topics, right? I mean, there's not too many master's theses that get such a broad scope of the field, right? Right from growing it to figuring out the taxonomy to like bioprospecting and working with mycologists all over the place to get samples. Really, yeah, it's a cool project.
Totally. And
Um,
this bioprospecting work was very complementary to the taxonomy stuff because my question was what species are producing this compound, and you can't really answer that question without knowing what species are these
there
things.
are. Yeah, yeah. Oh, what was there a reason it was Hericium originally? Was there like an industry sponsorship or something? I think I did see that in your thesis. There was some industry incentive to look at Hericium.
Yeah.
Was that correct?
Yeah, there was an industry sponsorship through MyTax, which was the company was looking at kind of maybe going in the direction of producing supplements based on functional mushrooms, and Hericium was part of that, and I was down. Yeah.
Yeah, I mean, lion's mane Hericium erinaceus, or the complex, is probably one or two for the biggest sellers in the mushroom supplement space, right? It's massive. The market is like literally multiple $1 billion marketplace for these things now. So it's not surprising that people want to create standardized products. And yeah, really interesting. But I mean, it's a fascinating species. But that was part of the driving force. Were you also interested in the species complex before, or was it kind of just like, hey, there's this project,
I
make it
had
happen?
I had no idea there was like taxonomical questions to answer when I went into the project.
Yeah,
But as I got
they
into it,
probably didn't either.
easy to get interested in it and totally immersed in the work, and then it becomes a fun game of okay, we got to go back through like hundreds of years of literature, looking at original descriptions of these species from the 1700s to see okay, this species or this picture matches most closely with this species in this region. That name was sanctioned, so we got to apply it and go from there in the modern era of molecular work.
Yeah, totally, totally. And then you were under Greg Thorne the whole time. How was that? How was working in Greg Thorne's lab? I mean, he's all over the place in mycology. I mean, he's described, I don't know, like hundreds of species, I'm sure, or at least lots. I mean, a lot. I think he did a lot of work with whole hen Bellina, like showing they're predatory. But he's done all sorts of interesting work in there. It must have been an interesting lab to be part of as somebody that's into mycology.
But yeah, Greg is such an inspirational person to work with. I'm not sure if you got to meet him
I
at
met him
college.
briefly. I was at the MSA conference. I met him briefly. I've had the honor of having some mushrooms identified by him, but that's about my relationship with him.
Yeah.
I'd love to spend more time though.
But yeah, any time with Greg is a good time, especially when you're out looking for mushrooms. But
Yeah,
yeah,
so much passion.
that experience was incredible, and I'm super grateful to have the experience to work with him and learn from him because yeah, he's got decades of experience, and his passion has not diminished whatsoever over the years.
Well, I find mycology, it's just bottomless, right? There's so much to learn. It's pretty accessible too. You just walk outside and you can start finding mushrooms, and the questions start almost right away because you're like, well, this looks like this, but it's kind of, there's this host interaction that I haven't seen before. There's this, it's kind of dry right now. What's it doing out? There's a million things to ask more questions. So it's like a bottomless reservoir of interest if you have the passion for it, right? So
Yeah,
and and then the other thing is, you could, there's a sort of somatic sense to it because a lot of times, I mean, the biggest, most of the mycologists I know are also big mycophagists. They love eating mushrooms, which makes it like a treasure hunt every time you go out too, right? So one for the scientific samples, one for the cook pot, right? It's kind of an interesting
yeah, yeah.
field in that.
There's no limit of reasons to get into mycology, and even if you just have your phone on you taking pictures of
Yeah.
cool looking mushrooms you don't know anything about,
Yeah.
and you post them on iNaturalist, maybe you get some insight as to what they are, and then that can trigger a lifetime of curiosity and interest.
Yeah, yeah. So okay, let's talk about so you go in, you think it's going to be a biofermentation project. You start reviewing the literature, you realize there's some confusion. You get some samples. Do you start just with ITS sequence? Is that what
Yeah.
I should maybe explain what barcoding is just to anybody that doesn't know, but and correct me, Julian, because you've probably done this more than I have. But basically, you're looking at a conserved sequence of DNA in the fungi. So ITS, I think it's intergenomic transcribed spacer region in the ribosomal subunit, which is just something that all fungi have, and it's conserved because it's extremely important for survival. So over evolutionary time, those small changes accumulate. So you can kind of use this sequence to compare, how distantly related fungi are, and it's the standard one for fungi, ITS. But that I imagine that's what you started with is just ITS.
Yeah,
Is that correct?
I think most sequencing projects start with ITS with fungi. It's, as you said, it's the barcoding gene, and it's easy to amplify. It's easy to read. But when we did it and we looked into the literature and started pulling sequences off GenBank, we realized that ITS is actually not that useful for delineating species in Hericium. For other groups, it's perfectly fine to attach a name to a species or a sample. But for Hericium, the genetic distances between closely related species is not huge for ITS. So we had to use other genes, protein coding genes. We used LSU, TEF1, and RPB2 as protein coding genes to kind of have that higher order taxonomic resolution.
And just to interrupt here and give some context, protein coding genes tend to be less conserved because there's more forgiveness for variation in them, right?
So protein coding genes are a little bit more conserved since they code for proteins. They're not.
More than the ITS.
Yeah. So
Okay.
because they code for proteins, any mutations
You
in a
lose
protein
function.
coding gene is going to have some effect on the organism. A lot of the times, the mutations are deleterious, killing the organism and not passing it on to the next generation. So they're highly conserved, and so they're better for higher order taxonomic comparisons. Whereas ITS doesn't code for anything; it's free to mutate over and over and
It's a
over and over.
spacer region between these important, yeah, so I explained it wrong. It's a spacer region between these essential things, so it's always there and it can actually evolve and change versus the actual coding regions can't. Yeah, okay, that
Exactly.
makes
And
sense.
the key thing with the ITS region is that it's in between two highly conserved regions of DNA. So it's in every fungal cell, but it's not going
But
to
it's not
have any
essential. Code's
effect
not
on the
essential.
organism as it mutates.
Yeah, yeah, cool,
Yeah, so we
cool.
did
Okay, that
a multi-locus analysis using these four genes. It was the first one to be done on Hericium. It's being used all the time now. And a lot of studies are going beyond multi-locus analysis to doing whole genomes.
full
But
genome.
yeah,
Yeah, I mean, and as the prices drop, I guess that's the way a lot of it's going to go.
yeah, and I'm super excited to see the next whole genome study with Hericium to, either suggest other, maybe we were wrong, and maybe the whole genome says something different than what the four genes that we found did. But yeah, we were fortunate to get good sequences from all four genes across the genus.
That's awesome. That's awesome. Was there any holdouts in that regard, or just it pretty much all worked when you went for it, or was there some species that were more challenging than others?
Yeah, I mean, some samples are more difficult to amplify than others for whatever reason. I think anybody who's done some genomics work can attest to that. But it's there's only so much time and so many resources to do an extraction over and over and over again. But for the most part, we were pretty lucky that they all amplified pretty well. The tricky thing was that on GenBank, there's not a whole lot of these protein coding gene sequences available. A lot of it is ITS, and of those, most of it is junk. Like I think I've seen some estimates that like 50% of up to 50% of sequences on GenBank are misidentified or improperly labeled. So
And they're not tied to a specimen. They're just sort of floating out there with from a who knows what a lot of times, right?
yeah,
That's
yeah, they don't have any like information of where this was collected or what organism it's come from. Maybe it was from like a mushroom breeder or a strain supplier, which is interesting, but it's not really useful to genetic studies because if a mushroom grower is breeding a species or a
that he
sample
got
from
from
you know Thailand with a sample
Asia
from
or yeah,
Mexico, it doesn't
exactly.
really represent a true evolutionary
Yeah.
event.
No. Yeah, no, for sure. Yeah, it's really fascinating. So I mean, we'll go into the five lineages a little bit. So basically, most people when they hear Hericium, they think lion's mane, right? Which is the one that is most commonly cultivated, and it's the one. Well, I mean, Hericium erinaceus was the name attributed to the 1990s Japanese studies finding these erinicins, finding the hericinones. It's the name attributed to all these events, but it's more complex than that. So there's five lineages, is what you're saying. There may be more, but at least five. And did you identify a number of species under all these lineages? Was it about 17 worldwide or something like that, I think.
Yeah, depending on your interpretation of the literature and like your interpretation of Index Fungorum, for instance, which is a good starting point for taxonomy or names at least. There's about 17 to 20 species that are commonly accepted. The five that were the ones we discussed in North America are Hericium aviatus, Hericium serratum, Coralloides, Americanum, now Carolinense and Oregonense, and I think I didn't miss any there.
No, I think that's it. So it's five lineages, six species in North America. That's kind of right, or that's kind of what you described, right? So the Carolense and Organense are they're ones that you named during your thesis work, and they're both under the Hericium erinaceus complex. And Organense is on the west coast. I think you identified it mostly on Gary Oak out here. Chorus Gariana. But it's funny because I know I live in Victoria, British Columbia, so I actually find some of these once in a while, and I usually find them on big leaf maple, even more than Gary Oak. Gary Oak savanna tends to be very dry, so your window for finding them is pretty narrow. But where there's big leaf maple, it tends to be wetter areas, and they seem to produce quite well too. I know you didn't have a sample from them. I'm assuming it's the same species though, because they're a hardwood lover.
Probably,
It'd
yeah.
be interesting to sequence. It'd be interesting to sequence. And then Carolense is the East Coast species, right? So running right up and down the East Coast, up into Ontario, down all the way to Florida, or do we
Yeah,
know if it goes that far down?
yeah. The holotype that we assigned was from Maryland on a species of oak, but it's probably pretty widespread. I mean, if you look at iNaturalist sightings, there's not a whole lot actually from Canada. Even when we're like, if we're considering this as Erinaceus generally, there's not a whole lot of Erinaceus like that we can be confident in from Canada, at least on iNaturalist. There's a few that might be, but they're mainly in the US. And Carolinense is probably more restricted to the East Coast.
Yeah,
And
yeah,
then
interesting. I mean,
the third lineage in that complex is Asiaticum, which yeah, the holotype we took from Japan, but it's probably pretty widespread.
yeah, and that likely is the species that a lot of this early work, out of Japan was on, just because it was what was available, and it's probably what is mostly commercially grown in Asia too, right?
For
I
sure.
mean, I
Yeah.
don't know. Did you
And
do any
probably commercially
sample?
grown here as well, because
Yeah,
I mean, when
a lot of this.
a lot of this work was starting to come over from Asia to North America, they were probably getting their cultures from Asian labs, so it's
Yeah.
probably Asiaticum.
Interesting. Really fascinating. I wonder if there's any crossbreeding. I mean, I know that's not something you did in your study, but I wonder if it could outbreed with some of our local species. I mean, really interesting from a conservation perspective. I know golden oysters are running rampant, especially on the East Coast. But it'd be interesting if this Hericium asiaticum was actually also spreading around. Due to cultivation. I mean, that'd be really fascinating to see. I mean, you need widespread sequencing efforts. I think, and like you say, people are a little bit protective of their Hericium finds. I'm a big iNaturalist user, but I don't upload my Hericium finds because I don't want other people to know where they are, right?
Yeah, yeah, it's understandable.
So I'm probably a bad. The reason there isn't a lot is because a lot of people finding these things want to eat them, and they don't want everybody to know where they are. but,
Totally, yeah. And yeah,
interesting.
the question of like escaping cultivation into natural environments is a valid one. I think, yeah, as you mentioned, the golden oyster phenomenon that we're seeing is, I mean, it's still in its early stages, yet to be seen how much of an effect it has on local populations. But
Yeah.
with respect to the Erinaceus group,
It'd be a
this
lot more cryptic because visually they look very similar,
well,
right?
that's the thing, right? There's not a huge amount or any really morphological distinction that we've identified between the species, and they're interfertile so they can breed and hybridize. So that's going to erode a lot of the genetic barriers between the species that makes future work a little bit more difficult. But this is the world we live in, and it's kind of just this is what we have to work with.
Reality. Yeah, it's the reality. Oh, it's just an interesting question. So let's talk about the species just for a minute that don't get called lion's mane. So there's the four other lineages. So there's Hericium americanum. Do you know what the common name of that is? I don't even know. I mean, they call them
I've heard
bear's
it's
head.
called bear's head
Yeah, I've
primarily.
heard Hericium abetus called that too. So I don't know, like lion, like they call them all sorts of different things. But Hericium americanum, that mostly a conifer species,
Americanum
or
is a
is
hardwood species.
it's a hardwood species?
Yeah,
Okay, and it's quite a frilly looking, like it looks quite different than the traditional lion's mane, right?
yeah, it's
Um
a little bit more branched. It's got long spines, similar to the lion's mane species, but compared to Coralloides, which we'll talk about in a second, Coralloides is more like it's branched, but the teeth are aligned more like the teeth on a comb, let's say.
yes,
A
yes,
little bit less compact and more frilly. It's a good way to put it.
yeah,
It's
yeah,
really distinctive. I think a lot of the confusion comes between Americanum and Erinaceus. Um, but
yeah, because it's a little bit more.
yeah, yeah. But the the Coralloides is quite distinct.
Yeah, and Coloroides is a hardwood species, or it's a conifer species, right? It's mostly a conifer associate.
Coralloides is hardwood as well. There's some reports of potentially in Europe. There's I think one sample that we looked
One
at that
sample
was on
from
conifers,
conifer.
but it
Okay,
was originally described from hardwoods, and we find it mostly on hardwoods here.
mostly on hardwoods. Okay, and I mean, there probably is cryptic species in this complex too. I know there's some. I know Danny Miller's website, which I use a lot when I'm identifying fungi. He does a lot of sequencing on the west coast primarily, but he thinks there's some cryptic species hidden in the Coloroides complex, very likely on the west coast. But I am sure it's the case all across. But it's not commonly cultivated. But really, it was one of the most interesting ones you found in your study, right? It was the big producer of erinisins, more so than the Arinaceous complex, right?
Yeah, totally. I think the Coralloides group has a lot of work to be done on it. As you say, it's a pretty variable genus, or excuse me, variable
Yeah,
species. Like it's
yeah.
probably a species complex with some cryptic species within it. We weren't able to nail
You
down
just need
exactly what
lots of
lineages
samples,
were
right?
from where,
Yeah,
if any.
yeah, yeah,
But you're right. It does produce erinosine A in substantial quantities. We found that through all of our analyses it produced the highest concentration of erinosine A, and
yeah,
even more so than the cultivated lion's mane mushroom. So I think if the industry, the mushroom growing industry, and the extracts and supplements industry moves more towards erinosine A specific targeted fermentation, Coralloides is a really good specimen or species to
yeah,
look at for that.
yeah. And so then the actual conifer associate, Hericium abetus, that's a west coast conifer species, and I think it's pretty broad in its host. Like I said, I found it a number of times, and usually slight elevation, and it seems to really like the mountain hemlock near the coast. Like as soon as you get up in elevation in sort of late summer, it's all over, and it is a true conifer associate though. It pretty much just likes conifer.
Hmm. Yeah,
And that's a unique North American species too. And so all those four lineages, they are pretty aggressive growers, right? And they're pretty widespread. I mean, Hericium abetus, I guess, is the least widespread, but it's still fairly widespread up and down the west coast. And then you get to Hericium serratum, if I'm pronouncing that right, serratum, and it's considered a rare lineage. But there's samples of it both in North America and in Europe as well, right?
yeah, it's a rare one. It was just moved to the genus in the 1980s. It was previously in Creolophus, but it's a rare one. It occurs, as you said, in boreal and mountainous regions in North America. We got a sample from Alberta. Luckily, actually, we contacted someone who had a sample that they posted to iNaturalist, and they sent it to us for analysis, which was amazing, and a huge shout out for the iNaturalist community there.
Yeah, it's
But that
amazing.
matched pretty identically to the species or to the samples that we found or that were available for Creolophus serratum in Europe. So that's a true species, and it's
Wow,
pretty widespread
interesting.
apparently.
Yeah. Interesting. Really fascinating. I've seen. I actually had the luck of getting a culture from a guy in Edmonton, and I think I should know. I should give him a shout out. But anyway, he was posting on one of the Alberta mushroom Facebook groups, but super slow growing culture. I think his name's Kensian Mycologist, which is just a neighborhood in Edmonton. But I could be wrong. But if somebody wants to reach out, I can find the guy if you want. But he has this culture, but super slow growing. And I never was able to fruit it. I was trying. I've grown lots of lion's mane just in my bathroom. I mean, it's very aggressive and grows quite well. And I've grown Hericium abetus too. But I've no luck with Ceratum. It seems to have pretty different lifestyle, or just a much more slow growing species, at least in the cultures I've had. Maybe there would be a culture out there if someone did some big bioprospecting work that would be aggressive enough to actually produce fruit. But it'd be really interesting. I don't think there's any chemistry work on the fruiting body of that species.
Not that I
Like
know
zero.
of. We weren't
There's
able
just not
to,
enough.
by the time we had kind of like wrapped up the bioprospecting experiments, that's when we got the sample, and we didn't
Yeah.
get living material, so it was or we didn't get living cultures, so we didn't get to include that in our bioprospecting analysis. But I'd be
Yeah.
curious to see the metabolic profile of that species.
Yeah, it'd be fascinating. It'd be fascinating because it does seem a little different than the other four, at least in its rarity, if nothing else, right?
In morphology as well.
Yeah. Yeah, it is. Yeah, it's a weird sort of shelf thing. I think it's considered endangered in Europe and a lot of countries, right? They consider. Well, I think even lion's mane is in some places. They actually have pretty strict rules about harvesting it. I think in the UK. I mean, I shouldn't be talking about this because I don't really know, but I know in North America it's just kind of a free for all. But in some places they do take these things a little bit more seriously. And the fruiting bodies, I mean, they recur year after year, but they're what produce the sexual spores, right? Which allow the species to spread and everything. So that there's always interesting discussions on when's the best time to harvest and when how to do it in a way that's ecologically sound. I'm not sure there's good answers to any of those questions, but they are out there, right?
Mhm. Yeah.
Yeah. I don't know. Do you have any thoughts about that one?
Yeah, I think just with the amount of lion's mane and hericium available for consumer markets that is out there, kind of it takes the pressure off while
Oh yeah.
harvesting
Wow.
for
Totally.
mass consumption. I don't
Yeah.
think the few individuals who are interested in going out to look for cryptic hericium species and
No.
try them, I don't think they're going to have a huge amount of damage to local populations. But that is to say, like, yes, harvest responsibly, and that is kind of like up to the interpretation of the harvester. I
Yeah.
don't think
It's a hard
these
one.
populations should be
Yeah.
decimated by individual harvesters. I think more of the conservation work should be targeted towards, whole habitat conservation and
Logging, forestry
logging,
practices.
exactly, industrial
Yeah.
pressures.
Yeah, and I mean that's any of these sort of saprobic slash parasite, like, because I mean these Hericium species they infect living trees with wounds primarily, right? And then the tree dies and they sort of digest it. I think some of the species probably are more parasitic and others are more saprobic. I mostly find Erinaceus on living big leaf maples, Acer macrophyllum. That's where I find it mostly. And once the tree dies, you maybe have a year or two to harvest it, and then it gets out competed by other saprobes that are more aggressive. I don't know. Is that kind of what you guys find on the East Coast too, or I mean. I know it grows saprobically very well in a sterile environment like a grow chamber and a lab and everything, but it doesn't seem to love living on dead wood in the wild, at least in my limited experience.
Yeah, so they're known as early colonizers of hardwoods, but I've found Americanum and coriolis on hardwoods that are in pretty advanced stages of decomposition, and that kind of goes against what the common understanding of their life cycle and lifestyle is. But yeah, the blur between, or the line between parasitic and saprotrophic is a little bit blurred with some of these species, and I don't think it's fully understood.
Yeah, yeah. No, it's a hard one. I mean, obviously it's sort of both, right? Like it obviously can infect a living tree with a wound, but it will survive in a dead tree too. So it doesn't need the tree to be alive, but it seems like a little bit better adapted maybe at living in a living tree than some of the more aggressive saprobes that are dominant saprobes, right? just an interesting observation. And but then I think Hericium colloides complex actually does better on dead wood sometimes too. I mean, I see those on lying down. Like the only times I've ever Abetus too. I found it mostly on like lying down dead logs versus Erinaceous complex. I almost always find on living trees or like freshly dead trees.
Yeah, when we were
Again, I'm
sampling,
an N of one. I haven't really studied this. I just, I eat all the ones I can, all the hericium I can find in the bush. But that's my experience with it.
totally, yeah. No, when we were sampling for specimens to include in this study, I think every single specimen that I found was on dead wood. None were found on living trees, but that's not to say they don't occur. And in Southern Ontario, where I was doing my sampling, there were no Erinaceous that we found. And as far as I know, no confirmed records of that species occurring in Southern Ontario, at least not sequenced.
Not genetically confirmed, at least. Like, so, iNaturalist confirmed. There must be some. I know somebody that found, it sent me a picture from actually on McMaster campus of something that looked like Erinaceous. I can probably find the picture somewhere. But I was like, well, that looks like it. I mean, they didn't sequence it or anything. I think they had it for dinner, but
Hey, it's entirely possible, yeah.
yeah, it'd be interesting. I mean, and that's the other problem with these things is they're out for a couple, like the fruiting body, the mushroom, like it's very hard to find because they're only out for a couple weeks of the whole year. They don't even fruit. They don't necessarily fruit every year. The ones that are easily, visible from trails and stuff tend to get grabbed by people who know what they're looking for. So you end up with a fairly limited, like you have to spend a lot of time actually to find trees that produce these things. And then, yeah, the tree maybe will produce it for a decade or something. And in that time, you got to get there for the 2 weeks of the year that it's visible or 3 weeks of the year, whatever it is, right? So it's kind of an interesting thing to find those fruiting bodies.
Yeah, it definitely makes the hunt fun.
But yeah, and when you find it, you
Rewarding.
feel like you're really you're onto something, right?
Absolutely.
Yeah. Okay. Well, let's start talking about the chemistry a little bit because I know did you publish anything on the chemistry? I don't think you did, right? It was just your thesis.
Yeah, it was just the chemistry work was only included in the thesis. The publication hasn't made it out yet.
Yeah. Oh, okay. Okay. So it's still in the progress. So let's not give anything away that's valuable. But was Aurinicin A the center molecule from the beginning, or you said you were bioprospecting, so looking at the whole array of chemistry? When did Aurinicin A become sort of central?
Aurinicine A was pretty central from the beginning.
Okay.
I
It's just because of that literature out of Japan and the stuff out of Taiwan. Is that kind of the
primarily, yeah. And when you take an HPLC analysis of these things, Aurinicine A is by and large the most prominent, at least Aurinacine. There's about 18 to 20 Aurinacines that have been discovered, and they were named in order A, B, C, super order.
yes?
Aurinacine A is like 300 times more concentrated in extracts than other Aurinacines. And it's the compound that actually has been studied in terms of its like medicinal properties on cell cultures as well as in human controlled studies and rat studies. So
Yeah.
Aurinicine A was definitely the target compound from the beginning.
Okay. Okay. And were you looking at other chemistry, or just sort of it was mostly you looked and you're like, hey, there's like way more Aurinicin A than everything else. Let's focus on that.
Yeah, and it was the most convenient too because there was published protocols on how to extract and isolate it and purify it. So I was like, I'm gonna take that and run with it.
Yeah, and at the time there was no standards of it, or the like super expensive standards, right? Is that the case?
The standards were like totally unaffordable for us. They were chemically synthesized, and the synthesis of Aurinicine A is like super labor intensive, super expensive.
I didn't even think you could synthesize Aurinicin A. I didn't even know you could.
Yeah,
I thought there was no direct path, but maybe they have some sort of precursor or something and modify it. Very interesting. So you had to produce enough biomass to get enough Aurinicin A to use it as a standard to actually analyze these things.
Yeah. We were. I think I took up every single 4-liter Erlenmeyer flask in the lab. I didn't leave any for anybody else, unfortunately. But we had to produce a lot of biomass to get that Aurinicin A standard, to which we can compare extracts against for all the other species that were included in the study.
Super fascinating. Yeah, oh, that's really interesting. So when you started, did you know these other North American species produced it, or it was pretty vague, right? Like the literature was kind of not like there's some maybe some a little bit of literature showing Hericium colloides, but were you expecting it to be like a genus wide uh, molecule, or was it that kind of surprising?
I had my suspicions. I mean, there's literature to suggest that the newly described species from New Zealand, Hericium nova zealandiae, was producing irinosine A. And a species from India produced irinosine A. And so I had my suspicions that this was a genus-wide phenomenon, and it kind of gave me a good starting point to target my study to grow the mycelium of all of these species, extract them, and then look for irinosine A. And yeah, we found that every species that we tested produced
Produce
irinosine
some.
A, and Hericium colloides, which is not a commonly cultivated species, produced the highest concentrations, at least under the conditions that we grew them in.
Yeah. And that's really interesting because the commercially grown species were not the highest producers. Whether those are, I mean, they're Hericium erinaceus complex, so they're probably Hericium. Oh, did you sequence these ones? Did you identify which these commercial strains were matched to, which lineage?
We had one commercial strain in our study. It came out to erinaceus from Europe, but that was the only commercial strain that we had access to.
Okay, okay. That's interesting. Was it from Europe, or could you find the lineage of how you got that strain? Because sometimes they're very storied, these commercial strains, depending on who you get them from. They're from some guy who from some other guy who had a friend in Japan that cloned something from a grocery store, or who knows, right? Like it's kind of funny sometimes how these strains came to be.
Yeah, no, the lineage of this particular sample was kind of cryptic. I was like given a plate from a friend, and he's like, "Oh, this is a lion's mane that's grown commercially." I'm not sure if he got it from a grocery store sample
Yeah,
or what. So I didn't trace the lineage of
yeah.
that one.
Did they actually talk to the guy growing it, or did they just clone it off a grocery store mushroom? It's pretty funny, right?
Yeah,
Yeah. Yeah, so I mean, really, the essential question here is, if they're trying to produce, especially the medicinal fungi industry, if they're trying to produce this specific molecule, did we standardize around the wrong species? Like, is it standardized to the wrong species of Hericium? Because I think you had, I can't remember what it is, but like about six-fold increase in production in the Hericium colloides cultures that you used under the conditions that you grew, at least.
hmm yeah. I think it really opens up the question of what species is best to cultivate for targeted production of irinosine A. I mean, the methods to grow and fruit lion's mane are well established, and people are comfortable and have their preferred cultures, and that's totally fine. I love that aspect of mycological cultivation. But if a company is going to be growing a bunch of mycelium to have irinosine A in it, it might be useful for them to look to other species in the genus to see if those are more potent, let's say.
Yeah, well, more potent and easier to grow biomass because Leucoides cultures tend to be very vigorous too. I mean, in my limited experience, so it's not like you're changing vigor for potency. You have something that's equally or more vigorous that's producing more of the bioactives of interest, right? So,
Totally. Yeah.
um,
And I think that's again where like the taxonomy work kind of like folds into this stuff is that with a
yeah,
better with a higher resolution of species names and lineages, you can better breed certain strains to maximize irinosine A production if that's what you're after.
yeah, yeah. So, with a six-fold increase in with these species, did you rerun any of these experiments just to see it? Because it's pretty extreme. Like, I mean, that's a huge fold change, right? It's
Yeah, we
almost an
did.
order of magnitude more, right?
Yeah, no, we didn't duplicate any of the species, or excuse me, didn't duplicate any of the studies. We did have, I think the sample size was an N of 5 for each of the samples that we ran. So that was the mean of those samples. I would
Yes.
I would encourage anybody else who's looking at this stuff and interested to run these samples again to verify or validate some of this work because it's a pretty marked increase in production, and
Yeah.
that's meaningful for a producer of this stuff.
Totally. Totally. And so, do you think there's a future where these other North American species are more cultivated? I mean, you don't see them in the marketplace really. I don't think people are growing them much except hobbyists at this point. But do you think there's a future where Hericium colroides becomes something that people actually cultivate and actually take as a supplement, or actually, or is the brand power behind lion's mane probably already too established and too strong? I mean, that'd be my fear. I mean, if you're a company trying to sell this product, you don't really want to have to educate people on a name if you don't have to, right? But it just maybe the education's around Arenisin A as opposed to lion's mane.
Yeah, I think that's exactly it. Like a lot of companies don't want to do the hard work of educating the consumer of like, hey, this is a new species that's edible and actually pretty good for you. And this compound is in it, which has all of these potential aspects. Like they're kind of riding on the legacy of people who have done that previously and attaching their name to a supplement, which
Yeah,
is totally fine, right? It's a lot of work. It's a lot of money to do that
how
stuff.
the industry works, right? It's so expensive to create a new brand around a product people don't know about. I mean,
Mhm.
so I understand, but if it's not the most potent one, you wonder if there's hope that it could become the most potent one, right?
Yeah, absolutely. And I think we're still at the beginning stages of breeding lion's mane for like the submerged liquid culture aspects of its biology. A lot of the breeding has been done for fruit body development and high yielding and uniform yielding, and that's great for the fruit body market. But I think in terms of the like mycelium with enriched Arenisine A compounds, I think having more breeding for that specific purpose might yield some brand new strains that we haven't seen yet.
Yeah, yeah, no, it seems very likely, right? So let's talk just a little bit about Arenisin A itself and why people are interested in it. So I think it was like we said initially described in the 90s in Japan, and the really exciting thing was it stimulated nerve growth factor, which is a neurite which causes neural differentiation in your brain, right? In mammalian brains. I think they probably started with cell culture, and then they put it in rats, and then there's been some human studies now, which it's very fascinating, and there's not a ton of molecules that can cross the blood-brain barrier and then do that. And to find one in a well-known medicinal fungi, quite unique, right? And I want to flag this too while we're talking about it, but lion's mane traditionally was not used for brain health. It was used, I think, for digestive health primarily, right? But it has a long history of use, but mostly around digestion.
Yeah, yeah, it was. I think the earliest documented records of its use are like 6th century China in the Sui dynasty. Um, but it was, yeah, primarily used for gastric ailments and to improve digestion. And only recently in the 90s when Kawageishi discovered that they produce hericinones and arenosines, and those compounds have an effect on nerve growth factor production in mammalian brains, did we flew into the cognitive and neuronal health aspects of this mushroom. And the key thing with Arenisine A is that as you said, it does cross the blood-brain barrier to stimulate nerve growth factor release in vivo, which is pretty unique. A lot of drug companies have tried to develop drugs that do a similar kind of mechanism, but a lot of the time they can't get the drug to cross the blood-brain barrier, or it's just too expensive to produce a drug for these things. And not a whole lot of drugs for Alzheimer's and Parkinson's disease are being developed, and it's expensive to do so.
No. Yeah, yeah. So this molecule has been linked to like you said, Alzheimer's, Parkinson's, stroke models. I think depression models. There's all these different systems that it seems to have some effect on, right? Which like you say, it's a challenge for big pharma even to create molecules that do these kind of things. And here's this natural molecule in a fungi that's been used since the 6th century, that seems to do this, right? Not the tissue that was used in the 6th century. I mean, it would have been all fruiting body then, just because they didn't have a good way to produce the 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 mycelium in like a sustainable way. I don't think until much more recently. But yeah, what a fascinating find, and so very interesting. And then it just sort of spawns out. But the big bottleneck around it is like consistent production of like an effective dose of this molecule, right? There's not. I mean, I really only know of one company out of Taiwan doing this. And now there's a number of companies starting to sell products using, I think, this starter material. I won't name names, just whatever. But so that you can find like there, I just saw advertised an Arena Prime product. There's an Arena Max product. So there is some products coming to the market, but they're all off this cultivation, this liquid cultivation going on in Taiwan. And then, as far as I know, nobody's doing it in North America. And that being said, they're using the local Hericium, very likely Hericium asiaticum, which might not even be the best producer, right? Though they've bred it, they've done whatever they needed to do, to create the best culture conditions to produce the most arenisins they possibly could. But still, it might be interesting if they had picked a species that's naturally a better producer, maybe the yield would go up substantially, right?
Yeah, it could be. And in whatever species or strain that they end up using, the optimization of arenosine A production is going to have to be strain specific. That's what we found in our studies. It's not like you can publish a recipe for, put this, and this in the broth and you'll get your arenosine A at this concentration. That's really strain and, case specific. So yeah, it remains to be seen what the industry is going to do in terms of arenosine A production in the supplement market. And again, I just want to like flag, like I think there's obviously there's a lot of research on this compound and on these mushrooms in terms of potentially helping with some cognitive function or some biological markers. I don't think it's a cure-all necessarily, but it doesn't hurt to have it in your diet. I don't think if you can tolerate it.
Yeah, yeah, no, yeah, it's super fascinating. I mean, I guess it brings up the question of like, naturally, if you eat these things that you wild harvest, you probably eat them, a couple times a year in the fall or the spring, and that's it too. You don't eat them every day, right? So there's not probably a lot of history of everyday exposure to these things, but obviously with supplements, that's a pretty different, a pretty different story, right?
Yeah.
So who knows what the long term effects are? But I mean, it seems like well, certainly that's what happens with the supplement space is you get to do these these large studies on a huge population of people. I mean, the one sad thing is it isn't tracked that well, right? Because it's just sort of whoever's taking it, right? But interesting. It's definitely never happened before where there's thousands of people all over the world, if not hundreds of thousands of people taking these things almost daily, right?
Yeah, and I think anecdotal evidence is anecdotal evidence, but it does have some validity in terms of
Oh yeah,
informing the future progression of the industry. I mean, you would know this stuff more than I do, being in the space that you're in. But
yeah,
yeah, I
yeah.
mean, I'm still interested in growing lion's mane. I'm still interested in taking lion's mane.
I eat it. I eat it all the time too. And I'm not trying to besmirch it, but I do think it's just interesting how these things play out, right? It's like there isn't really a history of daily use of these things, but here we are.
Here we
Here
are,
we are.
exactly. It begs more studies.
Yeah, yeah, exactly. That's what it needs, right? But it's just who's going to fund them and where's the human capital and the money going to come from to do it, right? But no, it's super fascinating. Um, so these cyanthin diterpenoids, I'm probably saying that right. Cyathin? Is that how you
However
say
you
it?
want to say it, however
Yeah, right. I'll just say it wrong and we'll pretend we know what we're talking about. But arenicin and the chemical complex these arinicins are in, they're actually found fairly widespread. Like I think Cyathus is a genus of bird's nest fungi. I don't know if you ever. I mean, I'm just describing for people listening, but they're little cup fungi with little like fake eggs in them. They're the bird's nest fungi. Well, they look like little bird's nests, right? And that was where it was actually these molecules, this class of molecules was originally described from, and then they were found to be in all these other species. But they're widespread in the fungal kingdom, and we already talked about like who knows what they do, but they probably provide that they must provide some competitive advantage because it's an expensive pathway, whether that's defensive, communication, or some sort of cooperative function. Who knows? But
Yeah.
yeah, really, really fascinating.
Yeah, I think they're found in like 5 species that I'm aware of. Sarcodon, Phelodon, Cyathus, Laxitextum. These are fungi in different families, in different orders.
Yeah, they're not similar. Like
They're,
Sarcodon and Phelodon are both like toothed fungi that grow on the ground, right? Mycorrhizal species.
yeah, they're super
Cyathus,
distantly related. So this is
yeah,
probably a case of independent evolution of these biosynthetic pathways.
which is really fascinating because it means they're definitely doing something important. If these different like very distal species of fungi are producing these things, right?
Yeah, they're not cheap to produce, and they wouldn't have stuck around so long if they weren't absolutely critical.
Doing something. Yeah, exactly. Yeah. Um, Yeah, so I don't know if you want to talk about your specific fermentation stuff. Maybe that's coming out in your publication. I know it's a lot of it's in your thesis, but I don't want to don't want to jump the gun if that. But you were looking at different carbon sources and adding oils and then agitating the actual samples to try and see how those things affected growth.
Yeah.
Did you find any specific input really change things?
So what we ended up doing was we bioprospected all of the strains we had access to and chose the strain that produced the highest Erinacine A from each species and developed optimization studies for the liquid culture for each of those species. And what we ended up doing was we looked at, okay, what are the different parameters that we could try to look at? And in liquid culture, there's kind of like endless numbers of parameters that you could choose. There's carbon sources, nitrogen sources, different minerals, different agitation regimes, temperatures, light, dark, it's kind of endless. And in the context of a 2-year master's thesis, you kind of have to kind of pull the reins back and decide on, a few different ones. And we ended up looking at carbon sources, nitrogen sources, agitation regime, and the use of plant oils, because the theory was, fatty acids could be integrated into the cell membranes, which could increase absorption. But all this is to say that what we found is that they didn't unanimously respond to any inputs in the same way. They often showed opposite trends where let's say one carbon source increased production for one species and it decreased production for another relative to a control. So it really is strain specific. You really have to design your liquid culture component based on the strain that you have access to or the strain that you're trying to grow.
Yeah, that's interesting. I mean, it's challenging, but it's very interesting. I know there's actually some published work, I think, out of the Helmholtz Institute in Germany around maximizing the production of Erinacin C in Hericium arenaceus, which they're in Europe, so maybe it is the true Hericium arenaceus. But did you look at some of that work too? I don't remember what they did. They used some sort of like oatmeal, and then they added some minerals like zinc and stuff, and they found that it substantially increased the production of Erinacin C.
I kind of, yeah, I
There's
I remember,
I think you maybe cited that.
yeah, I looked at a bunch of studies that did similar things. That one I included as well, and kind of synthesized what are the main parameters that these studies are using to.
And it wasn't the oatmeal.
Yeah, it wasn't oatmeal.
Yeah. So no, it's interesting. So you came up with just a plan that was manageable in the scope of your thesis work, and you just went for it. And then somewhat surprisingly, I guess you found that your Hericium coloroides strain was the best producer by like almost an order of magnitude.
Yeah.
Really interesting.
That was really interesting and kind of exciting in terms of showing me at least that we're still at the beginning stages of this type of work. We still, obviously we're at the beginning stages of bringing mycelium products or optimizing mycelium for Erinacine A production, let's say, to the masses. And within that field, there's still a lot of work to be done with what strains we're using, what species we're using, and from where and how we're growing them.
Yeah. So do you think, like, especially around medicinal mushrooms, do you think the future is more in like these big fermentation companies than it is in traditional cultivation? Because to create standardized products, you almost need to do these things at scale with a specific strain that's been optimized, right? Like I in my mind, that's probably the way the future of these things are going. It starts with bioprospecting, then strain optimization. Maybe breeding programs could go under bioprospecting. So then strain optimization, and then just like being consistent and producing the same thing batch to batch to get an effective dose of these things, right?
Yeah. I think the future is wide open with that potential. the fruiting mushrooms aspect, I don't think it's going anywhere. I think there's always going to be a place for that for the culinary markets and for the medicinal markets. But as we've talked about, the like not just in Hericium but in other species as well, there's a differentiation of products that are produced, like bioproducts that are produced from the mycelium versus the fruiting body. And depending on where certain maybe medicinal studies show the optimized form of different species lies, the market's going to probably respond to that and say, okay, well, for this species, fruiting bodies are better, let's fruit them. For this species, liquid culture is best, let's grow 30,000 liters of this stuff.
Yeah, yeah. Yeah, no, I think it's the future. And I think, well, and I think phylogeny kind of has to be the roadmap to the to the first arm of this, too, to some degree, right? Like, you
Hmm.
have to understand what your starting material is
Totally.
and how it compares to the other starting material. And I think that's really, like, the foundation of your work is, like, hey, like, we don't really know what the heck's going on here. There's a billion-dollar industry around just supplements, not even talking about the pharma potential of these things. And they don't even know, like, we're not sure what we're selling from batch to batch, right? To the species level, which, I mean, a species is a pretty distinctive thing.
Yeah. And I mean, I don't expect every mushroom grower to adopt the names that we proposed. Like, it's whatever makes sense to keep track of for every person makes sense to me. I'm not firmly in the camp of splitting
You're not a
this
splitter or a
a
grouper.
grouper. I think having a higher resolution of information with respect to what species we're calling what is can be useful, especially in the conservation aspect of mycology and in the industrial applications. But if somebody says, "Hey, I'm growing lion's mane. I've been growing lion's mane for 20 years. I'm not changing the names of what I'm using." I
No,
don't
and,
care.
and and again, it comes back to like informing your customer. Like, can you imagine if you have to tell them it's a different species than what you think is lion's mane, even though it's visually identical, right? Um,
Yeah,
yeah,
I don't
yeah,
think
no,
that's
no.
going to happen anytime soon.
No, me neither. Me neither. Yeah. So what? Well, we're kind of wrapping this up now. But what was the hard like of all this work that you did? What was the hardest part? Was it getting enough of the Arenicin A, or was it trying to get all these samples to do the taxonomy, or what? Was there any particular stumbling block that was particularly challenging?
Um, I think the analytical chemistry part of things was a little bit more challenging than others. I had done some organic chemistry prior to this work, um, and but this was all brand new information to me, and all of these techniques and instrumentation was, yeah, I had to learn from scratch. And I was lucky to have support around me and from people, more senior students and supervisors that were willing to teach me. But the nuances and intricacies of preparing samples for HPLC and mass spectrometry, and then subsequently analyzing those graphs and the data that they put out was challenging, but a great learning opportunity too.
Yes. Yeah. Totally. If you had like unlimited time and funding for this project, and was there something you would have done differently, or would you would have added on to the project?
Oh man, yeah, I would have loved to include more samples for the like maybe like a worldwide study, if that
Yeah.
was possible.
Yeah.
Whole
Yeah.
genome sequencing would be amazing, and I'm really excited for when that work does happen with Hericium, to see where it compares to the work that I did, and trying to include more parameters for optimization of liquid culture production of mycelium. I think that's where I was kind of disappointed that I didn't have more time and money to spend on, adding
More
different.
complex mixtures,
Exactly.
more
Exactly.
controls.
Yeah.
Were you just doing it all in flasks, like agitated flasks too, or did you have like little biofermenters?
No, I was just doing it in flasks. Anywhere from like 250 ml flasks up to 4-liter flasks.
Oh wow,
So
okay.
I was
And so just
small scale.
aeration through agitation.
Yeah, yeah, and that was what was available to me, but that's not representative of what's necessarily being done at large scale production facilities. This wasn't a bioreactor, and so this was what we found under the conditions that we grew it in. But that's not to say that someone else might find something different in different
Yeah,
conditions, and
yeah,
more than
yeah,
likely they would find something different.
yeah. No, totally, totally. Yeah, no, it's super fascinating. I mean, this just the surface has been scratched on fungal medicinal chemistry. Really, like there's so much chemistry in these things, and there's so much potential, but it's hard to access, right? And especially if you're looking at fruiting bodies that are there for 2 weeks a year, you got to know where they are almost before you start, right? It takes years to establish where these things are. And then the chemistry between the mycelium and the fruiting body is quite different. You can try and clone them, but the lab is a very different place than nature too, right? So some of these compounds are only produced under certain stresses. It seems like arenasin A is produced all the time though. Like it's constitutively produced, which is makes it a really good candidate for study. But I'm sure there's a host of rarer these compounds that would only be produced under certain stresses that nobody's looked at, and will anybody ever look at? It's hard to know, right? But some of them might be really interesting.
Yeah, yeah, I'm sure there's thousands of compounds that have some bioactivity profile on our biology that we haven't even identified, let
Yeah.
alone studied.
No, yeah, it's almost a bottomless reservoir, right?
Yeah, and
Yeah.
that's exciting
No,
and daunting, but that's uh, that's the fun part of mycology in our modern era is that we have the tools now to look at this stuff, and if you can,
deeper than ever before, right?
yeah,
But I mean, it's not to say that there isn't more to ask, and that's like someone like Greg Thorne, he's still excited, and he's been in the industry or in mycology for his whole life, and he's still excited, as he's matured in it. And there's no reason to ever lose hope in this space because there's just so much. There's just so
yeah,
much.
no, quite the opposite. I mean, it's beyond. I think it's gone beyond the borders of academia. This is a citizen science now that people are contributing really meaningful work just with an at-home lab or like a community space lab, and it's really made mycology accessible for anybody, whether you're associated with a university or doing it in your basement. I think everybody has something valuable to contribute and push and innovate within the space, and that part really, really excites me.
Yeah, super cool. Well, yeah, I don't know. Julian, that's kind of all I had for you. Was there any closing remarks you wanted to make about the space or about Hericium or your relationship with Hericium? Or
Yeah. Well, no, thanks so much for having me on, man. I really
yeah,
appreciate
no,
it.
I appreciate it. A
Fun to talk.
really fascinating talk, and it's such a it's $1 billion industry around this one species or this one species complex of fungi, and people, it's amazing that this work is being done, and like you really shone a light in a way that nobody else has done on this $1 billion industry, and like what are we actually consuming? What are we actually eating? Is this giving us the chemistry we're paying for, right?
Yeah.
So fascinating.
Yeah. It's a fun time to be in mycology, and I can't wait to see what the future holds with
The
respect
next
to
chapter.
Erysium and with respect to other species. Yeah,
Yeah. Yeah.
I'm stoked every day on it for sure.
Yeah. Is Hericium your favorite species or favorite genus of fungi, or do you have another that you like better?
Oh, a favorite for what? I mean, for eating, maitake is my favorite for sure.
Maitake,
Yeah.
yeah. Oh, maitake is so
But
good. Yeah.
yeah, I mean, I think Harissimo will always hold a special place in my heart, whether I stop working with it or not. It's a fun one, but I'm not restricted in my love for fungi to just a couple of genera. I think there's fascinating work happening in even the microfungi and in filamentous fungi, non-mushroom forming fungi as well.
Yeah. Yeah. No, absolutely. Yeah. And macrofungi get more attention just because they're so much more picturesque, but the microfungi is where there's so much more diversity, right? The ones that stay microscopic. But yeah, super cool. Thank you so much for your time, Julian. It was a pleasure, and I think we got some really good content here that people will be interested in. So
Thanks so much for
that's
having me on.
great.
I really enjoyed this talk.
Okay, take care, Julian. We'll talk soon. Okay, bye bye. Oh, thanks, Lucas. Okay, bye bye.